In this video, I'm going to talk about the process of fertilization. Fertilization occurs when an oocyte combines its nucleus with that of the sperm. Because each of these reproductive cells are haploid and only contain half the genetic material needed to form a full human being, the combination forms a diploid cell. So this process is going to generate what we call a zygote, which is a single cell that contains all the genetic material to form a human, half of which is coming from each parent. So let's talk about the process as the sperm reach the site of the oocyte where fertilization can occur. So fertilization depends on having a large number of sperm because most are not going to survive the journey. We're going to start with over 100 million germ sperms, and that are released into the vagina during ejaculations. Millions of these quickly get destroyed by the acidic environment of the vagina. The seminal fluid does provide some buffering that helps to protect the sperm, but still you're losing hundreds of millions of sperm almost immediately. Millions of sperm then are also going to be prevented from entering the uterus by cervical mucus. The thick cervical mucus essentially traps the sperm and doesn't let them in. So the sperm that does continue on, a fraction of them, a significant fraction of them, will be consumed by phagocytic uterine leukocytes that are going to take up and destroy the sperm. Thus, typically only a few thousand sperm are typically going to reach the uterine tubes, which is the most common site of fertilization. And really, that actually reach the oocyte, you're going to be looking at just dozens of sperm that typically make it to that. If the sperm do not immediately encounter an oocyte, they can survive in the uterine tube for 3 to 5 days. It only takes them about 30 minutes to 2 hours to get to the uterine tubes because they're helped along by uterine contractions. An oocyte, however, cannot live as long as a sperm. It survives independently only for about 24 hours after ovulation, which means that intercourse more than 1 day after ovulation usually will not result in a successful fertilization. One thing to note is during this travel, the sperm are going to be undergoing a process known as capacitation. The capacitation reaction is going to do a few things. So capacitation is going to increase the sperm motility, helping the sperm actually get to the oocyte, and it is also going to be involved in membrane thinning. So the membrane has, especially the head of the membrane, has cholesterol in it, and they remove that cholesterol, making it thinner so that it can release the contents of the acrosome and digest their way to the oocyte. In order for fertilization to occur, the sperm must complete the capacitation. So if sperm reach the oocyte before capacitation is complete, then it can't penetrate the thick outer cell layers. All right, upon ovulation, the oocyte is released from the ovary and swept into the uterine tube by the fimbriae of the oviduct. The fertilization most often occurs in the distal uterine tube because an unfertilized oocyte, as I mentioned, only survives about 24 hours, and it typically takes around 72 hours to get to the uterus. The ovulated cell is considered a secondary oocyte, um, and it's going to be surrounded by a few protective layers. So we have the outer layer, which is the corona radiata. It is made up of follicular or granulosa cells that formed around the oocyte in the ovary and remained after ovulation. Beneath this, we have the zona pellucida. This is a thick, transparent glycoprotein membrane that surrounds the oocyte's plasma membrane. As the oocyte moves through the uterine tube, it's going to encounter capacitated sperm that has survived. The sperm are going to be chemically attracted to the oocyte by substances that are being released by the corona radiata. To reach the oocyte, then the sperm has to get past both of these layers. And that is going to be where the acrosomal reaction is going to come into place. It helps to get through these two layers. The acrosomal reaction involves the sperm's release of their digestive enzymes. And as these enzymes digest a pathway through the zona pellucida, the sperm eventually are able to reach the oocyte. Ultimately, a single sperm is going to bind to receptors that are on the membrane of the oocyte, which are going to facilitate then the entry of that sperm, specifically its head and mid-piece that go into there. One thing to note is that the binding here is going to help with the acrosomal reaction, but some of them are going to spontaneously go through this acrosomal reaction process that helps to digest the matrix for other sperm. So really, fertilization, you oftentimes hear about it being the fastest sperm is the winning sperm. It's not always, and very rarely, in fact, is it the first sperm that gets there, because it's such a cooperative process that involves hundreds or dozens of sperm breaking down the protective layers for the one sperm to actually get in there. That's why if you have sperm loss, a low sperm count can result in male infertility. Even if you have some sperm that is still there, you need multiple sperm in order for this process to be successful. Once contact with the zona pellucida triggers the acrosomal reaction, the glycoprotein membrane of the zona pellucida expose the plasma membrane, as mentioned before, which is going to be how a single sperm. So notice here, as I mentioned, the sperm's plasma membrane is fusing. It's going to release its head and midpiece, and so you're going to have the sperm nucleus, which is known as the pronucleus that enters into it. Once the once the sperm has entered, you start undergoing what's known as a cortical reaction. So this is there to help prevent polyspermy. The cortical reaction is going to involve some proteins that are known as zonal inhibiting proteins and mucopolysaccharides that help to separate the membrane from the zona pellucida. So this is then going to form what is known as the fertilization membrane. This is going to prevent polyspermy from occurring, and this is basically to ensure that the cell does not end up being a triploid zygote with 3 chromosome sets or having more. The cortical reaction is also known as the slow block to polyspermy. There is something known as the fast block to polyspermy, but that is not something that happens in mammals to our knowledge. It's just believed that few enough sperm reach there, that you reach the membrane at that precise time, that the fast block is essentially not necessary, but you do need the slow block where the granules, so you can see those granules here inside the membrane, are being released, thus creating the fertilization membrane and preventing excess sperm from entering. All right, the process of fertilization can occur naturally, but science has made advancements such that we can have what is known as in vitro fertilization occur. So in vitro fertilization is an assisted reproductive technology that helps people get pregnant that normally would not be able to. So this allows individuals with a low sperm count, or that don't have normally have healthy eggs, or meaning that they can't reach the uterus for whatever reason. Maybe the uterine tubes are blocked or otherwise compromised. This can help those individuals become pregnant. So in this process, the eggs are collected after ovulation or directly from the follicles. Typically, this is a normal ovulation cycle only produces a single oocyte. So the treatment plan generally is to boost this by administering a short course of gonadotropins, meaning that you have follicle stimulating hormone and luteinizing hormone, or analogs to them, which trigger ovulation, allowing 10 to 20 oocytes to be collected instead of just one. Right before the ova would be released from the ovary, they are harvested using ultrasound-guided oocyte revival, where the physician uses ultrasound to visualize mature follicles and aspirates out the follicle using a syringe. Simultaneously, sperm is going to be obtained from the male partner or possibly from a sperm bank. The sperm does need to be prepared. One thing that happens is that they wash the sperm to remove seminal fluid because seminal fluid contains a peptide that in high concentration prevents capacitation of the sperm. So the sperm sample is also going to be concentrated, thus increasing the sperm count per milliliter. Both the collected eggs and sperm are going to be mixed in a petri dish, which allows fertilization to occur. The ideal ratio for this is about 75,000 sperm to one egg. But if for some reason the sperm are still having trouble with the fertilization process, the sperm can be directly injected into there. So the process here, yes, the sperm are going to be capacitated, but they're still not optimally prepared to fertilize the oocytes. Thus the reason why we generally have to concentrate them, even though in the actual in vivo, the situation would be that the oocyte is only going to be met with a few dozen to hundreds of sperm and not thousands. The fertilized embryos are going to be incubated until they reach their 8-cell stage, or the blastocyst stage. This is going to result, if we harvest them then at this point, in a more successful pregnancy rate. Then we transfer the uterus, or transfer the embryos to a woman's uterus using a catheter. After implantation occurs, the zygote should develop normally, just if it had been fertilized in the female's oviduct, because the principles are still there. So that was an overview of the process of fertilization. In the next video, I'm going to be talking a little bit about what is going on during development and what those changes to the organism as it develops is going to be. So I'll see you next time.